Hello! Welcome
Hi.
to Microbiology Journal Club. I'm your host, Faz. This is my co-host, Danny Chan, and this week we're going to take a deep dive into one of the really interesting microbiology papers that has been published. And this week we're going to be looking at mutations. Uh,
hmm Yeah, welcome to the program for those who are watching.
yeah,
We've been trying to do this for a while now, where we on every other week we sort of look at a broad view of different papers that we do that are in the news, and then on the following week we choose one during that week, and in the following week we dive into each figure. And today is one of those ones where we dive in. Yeah, lots of mutations in this one.
yeah. So the big question
And
of
so
sorry, do you want
Oh no, I was just gonna say this is something that like the beginning of this story is like maybe at the very beginning of this pandemic, right? And so it's sort of taken some time to develop that story and to see the data from it. Yeah, it's about this D614G mutation.
Yeah. So yeah, this is something that emerged quite early on. So there's this organization called the Global Initiative for Sharing Data on Avian Influenza Disease, who's been collecting genomes about around the SARS-CoV-2, like throughout the. So one thing that's amazing about this entire thing is that they've been collecting like every time well samples of people who haven't been infected with have been infected with SARS-CoV-2, and then analyzing that genetic code. Um,
Hmm.
and one of the mutations was this D4. So the original version of SARS is the D614, and new and improved version apparently is G614. Now this,
Yes.
yeah,
Yeah, that organization you mentioned, they have a website, right? And you can like visualize the mutations, I think,
yeah,
on that website. Is
yeah.
that
You can
okay?
access all these genomes yourself if you want to do an analysis and see some of
Yeah.
the big important mutations. Uh,
hmm And
uh,
that's sort of something you mentioned that was the A in that stand for avian because this is like a flu
yeah,
organization.
a flu organization. So they because nobody was caring about SARS-CoV-2 for a while because they didn't know it existed. But
Yeah.
avian influenza has been this looming and impending threat for such a long time that they've created this infrastructure for it. And the thing is, it can be
Yeah.
used to share data on multiple pandemic viruses, and that's what they've been doing. Um,
Yeah.
so like
Yeah, I think that that's maybe important to remind people of. It's like, we only in science like you only see what you're looking for, right? And
yeah,
like because we know flu is this seasonal, right, disease that sweeps the globe. I guess it's never like all at the same time. It's like different seasons in different countries. But there's a sort of relatively robust network of surveillance that surrounds it. And I guess it's being deployed against this virus as well.
yeah. And so the mutation appeared, but the question is lots of SARS-CoV-2 gets a lot of mutations, but so I'm gonna pull up that first
Yeah, we
slide.
should throw up the first slide, I guess.
Yeah. So the big question is, are they significant? Or how do you know whether a mutation actually is going to lead to a worse virus? Because lots of mutations just are neutral.
Yeah. Yeah. So in this figure, they're looking at four proteins: the spike, the envelope, the membrane, and the nucleocapsid. And they have tons of sequences of all the different viruses. And they lay out that amino acid sequence on the x-axis. And then on the y, we're seeing like the frequency of those mutations, I think. Concurrent, yeah,
Yeah.
variants. How often they see those variants. And I guess useful for this idea of does it even mean anything? They have the color scheme here, right? Where the red dots are amino acid changing and the blue dots are silent. So it's possible, right, to have a mutation that doesn't sit in a coding sequence, or maybe in that coding sequence it doesn't make a reasonable change, right? Codons
Right.
have some redundancy in them.
Yeah.
Uh, and even in this data, I think the D614G variant is present.
Yeah. So you can
Mhm.
see like it's so in terms of frequency, I can see it in I can't read
It's
in the uh
yeah, it's panel A in the spike. It's kind of in the middle. You can see it's like it's the highest. it's one of the highest dots they have.
Yeah,
They annotated it. Um,
yeah.
and yeah, so even back at this point, I don't remember what month this was. Ooh, do I have
Oh,
the month
this
for this
was I
one?
think in May or April or May, maybe around that time.
Yeah.
Um, and there was a hint, a couple of about like default D614G, but because it's appearing a lot, even in here you can see that there's a very high frequency,
Mhm.
and you probably might see more about all some of the other mutations that are name checked here, like N4 N43K or something like that. But yeah, well,
N43K. Yeah, that's also on the spike protein.
yeah. So we probably gonna the ones on the spike we're gonna especially see, and also like this is important like for the latest news because I don't know if you heard about that mink outbreak that recently
Hmm.
happened because
Yes.
the thing about mink is that
Caused the culling of a lot of minks.
yeah, the culling of a lot. We don't have a lot of data right now, but that is an example of where they found a new mutation coming out through minks, and they kind of found out immediately. They just came to a decision immediately that it's a dangerous strain and that we need to limit it as much as possible. Well, maybe not dangerous, but there's something that they want to limit the effect of because
Yeah, the potential of seeing things come to prominence like that.
yeah,
I mean, I think that that's important because, well, I mean, flu has a reservoir inside of other animals,
yes,
right? So it could be something like that. Like I could imagine it's a good reason to remove reservoirs. You don't want right? Like let's say we're successful in suppressing, right, virus around, and then it just jumps back once again,
yeah.
right, at when it's done. Yeah, that's an important thing. So wait, but they didn't report anything on I haven't found any
So
specific mutations coming
they
from the mink.
they they reported the mutations on their Twitter feed. Uh,
Okay,
so
Twitter data.
So that's the difficult thing. And I did check, and none of those mutations are name checked here, but there's like a couple of deletions. And the main concern isn't so much that it causes it to be more infectious, but more that it is very different from. So the areas where there are deletions are areas where the Regeneron antibodies would attach, or other
Mm,
antibodies are similar to humans. So they're worried that not so much will be more infectious, but more that it will mean that lots of our treatments to normal SARS-CoV-2 wouldn't work. So the antibody
sure.
treatments and potentially even the vaccine treatments might not work. So that's why they want to limit it. But
Right. And that could be a dangerous parental strain, right? Potentially. So like,
yeah,
right, the idea is that it's setting up for like if it could shift back into humans, right? That strain would be those daughters or those children, right, that come from
yeah.
the parental are might be dangerous. Yeah.
But that's but again, we don't know whether that would actually be as infectious to humans as it would be to mink
Yeah,
because
absolutely. And I mean, I think that like what I liked about reading this paper in context with everything, right, is that like this is the length of time, right, it takes to sort of understand what's going on. And
yeah,
policy decisions have to happen so much faster. Like policy can't happen on the scale of, right, tweets, I guess,
yeah,
right? Like as soon as scientists know and they can publicize it a little bit, then executive decision is made, right, by the bodies that make those decisions and say, well, what are minks anyways worth to us, right? Might
oh
as
yeah,
well kill them all so we prevent this potential danger.
and
But that's way before knowing, right, what that potential is.
yeah. I'm pretty sure they were planning on banning mink farms anyway. So this is like one of those
Mm,
like kind of extra ones where they go, yeah, why not? Let's just accelerate that. So
totally. Totally.
yeah,
Yeah, so anyways, in this one, this is they first identified that it's a rather frequent mutation.
yeah,
And then if we can go to the next slide,
yep,
um, there were studies that showed that this is like the first study that maybe put some function towards this, right? A potential function towards the mutation. Didn't directly show it, but it showed that over time, so like the initial outbreak, right, in China,
hmm,
started, I guess, in February here, at least monitoring. They have data from February.
yeah,
And that just has the D614.
yeah. So
Um, and in blue, they have the G614, and they're finding that in the later waves, that seemed to be what was more prominent.
yeah, and
Mhm.
that's it shows, especially in like different regions. Because in China it seems like it doesn't arrive as much prominence as it does in say Europe or even in Africa or North America.
Yeah.
But so
Yeah. So this is back in May, right? It's
yeah,
like, very much 2 months, I guess, into where we locked down at least, right? March was like the big month in the States. Probably should have happened sooner, right? Given some what was known. But yeah, all the way back then, this was observed. And I think in the media, I remember the media coverage was kind of scary, right? Because it's like
Yeah,
mutation, right? Mutation of this virus.
yeah. It's like
Um,
the X-Men.
and
You
I
don't.
don't know if, huh?
It's like the X-Men. You don't want to have mutation, a virus with eight light laser beams and stuff like that. That's
Yeah,
dangerous.
exactly. Send them to Genosha.
Yeah. That's a deep cut for X-Men fans, but yeah. Magneto is king. Yeah.
But yeah, like I think that's worrying for people to hear that word mutation.
Yeah.
But I think one of the things too to remember is that, and then people might like go to that website and look at the mutations over time, right, and see this phylogeny changing, and like that can be really scary. But again, remembering what do they actually do is like the big question,
Yeah.
and sort of trying to pin that towards some biological function. And I think we said this earlier in one of the streams. It's like you don't want to call them different strains of viruses because strains like imply that it has some different biological function, right?
Mm.
Isolated based on some sort of function. And maybe like talking about like sequence variants, right? And they
Mm.
are mutants. You can call them mutants, right?
Yeah.
It's just maybe the more precise way to talk about them. And during this time, we talked about this this
Mm.
paper like in our news recaps.
Mm.
And one of the things that we said when we looked at it was that this is very much just sequence data, right? And so we didn't really have anything else to say about it
Yeah.
um, at that time.
Yeah, there wasn't really much there, and want to be very careful to not like accentuate the kind of panic over it as well. So,
Absolutely.
because again, there's so many unknowns at that point, and we
Hmm.
didn't really have access to much more data. But now there is much more data out there, and people have been looking
Hmm.
into this in a lot more detail. So that's given us the opportunity to look back and see what the deal is with this new uh, G614, which used to be D. The nomenclature of this is kind of odd, but
Yeah, yeah. You say what it was and then what it became, right?
yeah.
That's how it goes. So like it
Yeah,
was D and it's becoming G.
yeah. So the nasty the one I don't say nasty one. The dominant one at the moment is a G614. But
Yeah,
the question is, how did it become dominant? Is it because it is genuinely uh,
more
uh,
infective or yeah.
or is it just that the only person who come out of China was the one who had that, and then that it got a head start in infecting people all over the world?
Yeah, so I mean that's our next slide is that example that you just said. In genetics, they call that a founder effect,
Yeah,
right? It's like where it's basically a bottleneck, right? And as you said, if one person left that had more of that particular virus, then they would only be spreading that particular virus, and that's just by chance,
yeah,
right? That's just the statistics of it all.
yeah. And if like China had like a really strong lockdown, then you would expect to see found effects like that, where because
Hmm.
the
Hmm.
smaller the
Hmm.
population that escapes, the less diverse the genetics is going to be, and so it's going. So some
Right.
strains are going to dominate just because, not necessarily because they're
Yeah.
bad.
Uh-huh. Yeah, and that was so like, looking at that last graph, like you can't really disentangle, right, a founder effect from, right, the peaks that you're seeing. But in that paper in May, they also provided something that I guess wasn't as easy to correlate with the founder effect, and it was a super slight effect because they have to sequence right? They had to do tons of viral titers here
Yeah,
to get at it. But they were showing like a small increase in viral titer.
yeah. Uh, so yeah, this is an interesting one. So yeah, they look at outpatients and inpatients and ICU patients, and then compared the viral titers and which one, whether
Yeah.
based on whether they had the G614 and D614.
Yeah. Well,
And
like, okay, well, let's look at A. It's A
A
first.
first,
A is the
okay,
one where they're showing the viral loads, and that's by CT. I don't think we talked about that as much, but like it's the raw, I suppose, data that comes off of the PCR, the
yeah.
RT-PCR test.
So these,
So lower CT means that you hit some right? Like you were able to amplify a bunch of copies sooner rather than later,
yeah,
meaning that there was more of it to begin with.
Yeah, and this is
Yeah.
talking only about the viral genome, so it doesn't actually tell you titers. So that's not actually necessarily the same thing as saying that there's more virus. It could just be something else that's happening in the cell that means that there's more genomes around.
Absolutely, absolutely. So again, this is like at in May, right? There's not very much to say about this. Like they're trying to point suggestions towards something that it could be, but
Yeah,
we're still quite uncertain.
and it seems interesting, but the thing is, like, there wasn't these. This could be saying it's more infectious, but the thing that we really care about at this point is does it cause more mortality? Is it associated with
Mhm.
greater death? And this is not what the paper showed. The paper didn't show any differences at all. And
Yeah, yeah. So yeah, in B, they actually looked at the hospitalization rates, and based on their stats, nothing really is different between these two.
yeah, I mean, the only reason there are some differences is because one of them is just much more common, but the actual ratios is uh don't show any significance.
Yeah, yeah. And so that was where the paper left it at that point, right? Like in May, it was like this came to prominence and they proposed, right, that it could be more infective. But when you read the data, the data to support that is not very strong.
Yeah. So so I guess the thing we'd want. So next slide, I'm just talking about the actual change itself. So it changes an
Mhm.
aspartic acid to a glycine. So these are two amino acids, and one of them has like this big like acid like negatively charged chain hanging off the end of it, and the other one has basically nothing.
Mhm.
So in terms of like proteins, it basically means that can change the way a protein interacts because firstly you're losing a big chunk of stuff that's in the way. So
Mhm.
but having a chunk of
So
stuff in the way is called
scaffolding maybe.
steric
Carbon
hindrance.
carbon
So
chains are like scaffolding.
yeah. So if you're playing with like a glycine Lego brick, it's going to be a little bit more flexible, and
Mhm.
but of course it doesn't have the option to like bind that this other negatively charged aspartate has. So
Yeah.
change that kind of change might, depending on where it is, can change how the structure of the protein acts. So
Mhm.
that brings us to the
Yeah,
next
and
slide.
I would say that the next work is done following up on that hypothesis,
Mm,
right? Saying that, okay, but this change seems like it might have something functional to do with it, right? We
right.
saw that slight thing inside of the populations where it might be more infective. So they trace it back to, they actually did cryo-EM, I think, in this paper, right? Looking at how the protein shape would change based on that one substitution. Um, it's really busy figure, but
Yeah.
they zoom in on it, right? In H and I, they zoom in on that interaction, right? Having the bulk sort of disappears, and without that bulk, it can't reach across this gap to
Yeah.
touch another part of the subunit. Um, and because it can't reach across that gap, then maybe there's a bit more flexibility for it to just flop around a little bit.
Yeah.
And they call that like the open conformation.
Yeah. That's and yeah.
um,
Sorry, I was going to say
is that all
something.
you want to say about that? I'm
No,
not.
I really completely forgot about it. But
Okay. So yeah, so potentially they have this more open confirmation, and then they want to relate it to actual infectivity. And so in cell culture, this group was able to go through and infect a lot of different types of cells with the mutant virus versus the wild type, I guess we'll call it. And they did show that you get a lot more infectivity. So more cells are infected on the dish with the same amount of virus that you initially give.
yeah, uh, yeah. So that's important. But the thing is, it's still in cell culture. So
Mm-hmm.
uh, sorry, I'm having some sound difficulties on my end. But yeah. What was I going to say? In terms of actually whether it has an effect in real terms is uh, an interesting point because we don't necessarily know whether it will. Cell cultures are very different from. So the example I'd give is like with E. coli. So if you're like working in a lab somewhere, you've worked with some form of E. coli that is good for genetic manipulation, and that's very easy to grow up in solutions and batches. But if you take a wild one that was taken from infection, they get very finicky. They're very. You really want to like give them a lot of. So the fact that
Yeah,
one of them will grow very fast in a petri dish doesn't necessarily mean that it'll do the same in humans. So, but
totally. I mean, and if you sequence those strains, right, they are different, right? And
Yeah,
we've talked about this before. Like cell culture, they've lived in dishes for some time, right? They have a lot of changes that allow them to live in dishes better than they are inside of organisms. And those changes could be the things that drive, right, this phenomena we're seeing of infectivity.
yeah,
So, there's always a push. We're always pushing to get closer to the reality of things with different models
yeah,
to try to figure out what's actually going on.
yeah. But of course, like given this data and everything else we've seen, like there's probably something there. And if
Yes.
I, yeah, because
Absolutely. I
yeah,
mean, that's why I sort of chose these two papers as the intro, right? Because we're sort of seeing how that information is building up over the course of scientific, right? So
yeah,
now, what, this is in June, I think, right? Or
yeah,
July, right? So May to July, right? Was from identification of something to moving into cell culture and feeling pretty confident that like there's something biologically relevant here, and we need to confirm it. They're saying it's infectivity, and they're pointing it towards this like structural idea. So enter our paper, I guess, right? Zhu
yeah,
et al. Now published as a preprint at the end of October. Um, they're going to give us more relevance by using all of the great tools that have been made since May to work with this virus and to work with models of disease in this virus and tell us, does this mutation actually do anything?
yeah. So the title is SARS-CoV-2 spike D614G variant confers enhanced replication and transmissibility. So the first thing they want to look at is uh, how well whether it actually changes the binding, the actual binding effect, and they use a technique called Biolayer interferometry, where where they basically have like a glass like stick that they have that coated in one of the parts of the so I think they coated it with either ACE2 or the variant of
Mm-hmm.
the SARS protein, and when the protein binds, uh, it thickens the layer slightly. So when they pass light through, it causes the waves of the light to come fall out of position, and because of
Out
that,
of phase. Yeah.
out of out of phase, and based on that, they can tell the degree of binding. So like when they're all bound, all the light forms back into a form of phase, but out. So I've
Yeah.
thrown up a diagram kind of demonstrating how it works from a
Mhm.
different paper about how,
Mm.
but we can just go ahead and see how it works in
Yeah. Yeah, I
here.
mean, it's interesting. I think we've talked about some techniques like this before.
Surface plasma
I know,
resonance
it's
was,
very loosely like SPR,
yeah,
except instead of looking at some weird quantum effect on the piece of gold, we're actually looking at it's like the distance, I guess, right? Between it's that very slight like separation between the space of these molecules as they bind to the layer. Probably easier to do is what I'm guessing because they you can test a lot of things is
yeah,
my guess. And again, you load up your favorite thing to capture on the surface. So yeah, a great way of looking at interaction.
yeah. And in this case, it kind of shows. They don't show it as dramatically as they could because the graph axes on these are actually very different. If you can see like kind of the shift
Mm.
and the
Yeah,
fact that they
one's the top is 0.12 and the other one's 0.2.
and even like the orange line in the D version is 75 nanomolecular, whereas the other one
Oh
is
yeah,
50. So actually, the difference is much more dramatic than they show. But they want to show you the separation of each line. I think that's why they
Mhm.
haven't put it all in the same graph and just shown you this trend. But this is a really
Sure.
big difference in comparison.
Mhm. Yeah, so the mutation really does have, and in some ways this is confirmation because it was also in that earlier paper where they looked at the open and close
Yeah,
that they also showed a similar binding difference. So this is one of the great things also about over time accumulating more data points, right? Like different techniques showing the same thing. So it's good, right? It's good that we're seeing this confirmation.
yeah. And the next they use a flow cytometry technique
Flow
where
cytometry, which we talked about before.
yeah, where again they look at like fluorescent signal based on
Mhm.
the S so this is the thing that is quite interesting. So I think the spike protein is separated into several sections, and they decided to look at just one of the sections, S1
Hmm.
section, just because I mean, looking at that connection, they wanted to check whether like it's due to the way it's bound to all the other parts of the spike protein in its own section, or whether it's partly to do with how it is in isolation.
Yeah. Yeah, because I think that was partially going back to the that structure that we saw, right? It's that the bridge, the mutation presumably mixes this bridge that it can make to another part of the subunit.
Yeah, so I've just pulled it up quickly. So you can see like the S2 is the one in yellow and S1 is the one in blue. And
Hmm.
so the connection between the S1 and the S2 subunit is like theorized to be a big portion of why it becomes a more open conformation. But I think in
Hmm.
here they decided to take a look at just the S1 on its own to see whether that is
Yes.
enough for it to be in that open conformation. Um,
Hmm.
so so
Well, actually, I mean, in some ways, it's or just enough to drive the phenomena of more binding,
yeah,
right? Because here in this version, I guess they wouldn't have like open versus closed necessarily because there's nothing to to keep it closed,
yeah.
right? Like these are just floating around with the mutation itself. Yeah. Hmm.
So yeah, in this case they have it so that when they bind, they both have like a fluorescent signal, and the degree of fluorescent signal indicates the level of binding. And
Yeah. Yeah, because I yeah, I think they're bound maybe to they probably they have a way of like marking it afterwards, right? Like these flow cytometry workflows, it's like you bind something to the cell and then bind something that glows to the thing that bounded the
yeah,
cell.
yeah, and
But
so
they see more fluorescently bound cells when they have this FC uh,
yeah,
with the 614G,
yeah,
but they don't see as much of a shift with the his tagged version.
yeah. This is where the paper kind of I didn't really understand why this was. I don't think I got really good explanation out of the paper for it. Because I mean the big thing is like they both say in both of them that there is a difference either way, but the difference is just much more dramatic with the FC confirmation, and
Yeah.
I'm not sure like what
I
that
mean,
means.
FC is a much larger thing,
Mm.
right? FC is like a much larger fragment that
Yeah,
would be attached to it. I mean, it's possible that they are trying to comment directly on the open versus closed idea,
right.
right? And like, oh, this is like bulkier, and maybe it can interact with another chunk, right? And so in that version, right, you see the difference a lot more versus when it's just a small tag. Yeah, I mean, it's not the most direct way, right, to get that question if they're trying to answer it, but it's something.
Yeah,
And I guess importantly, they are at least able to see a little bit of a difference, right, in affinity, yeah, between the two.
yeah. And the next slide,
Oh, so yeah, to set up the next set of experiments that they're going to do is that they have to make a mutant virus. And I don't think we've ever talked about making a mutant virus, but this is like also a classic way of testing. This is like for people who study infection all the time, right? Like we have two things we can vary mostly, right? Like you can vary the genetics on the host side or the genetics on the pathogen side. And so in this case, they want to make that mutation to the virus, and they do it by reconstituting the virus from an artificial chromosome. And I also read a paper recently. I think I even had it in the news once, but like you can do it with bacterial artificial chromosomes as well.
yeah,
It's just you need a really long piece of DNA because coronavirus is actually quite a long RNA virus. And so you need a really long piece of DNA, and then you can edit it however you want, your favorite editing methods. And then once you linearize it, it will make RNA that is the genome of the virus,
mm,
right, with all the edits that you need. And you can electroporate those, you can transform those into cells, and then those cells will essentially be infected
yeah,
by an artificial genome. So yeah, anyways, that's just a technique thing, but they're going to use this to create the mutation in the spike protein, and now they'll have two variants of a virus, one that they put through mutation and one that they didn't.
yeah. And they test it in vero cells. So they're from African green monkeys. They use quite a lot in SARS-CoV-2 research. And
We've
in
seen them so many times,
so many times.
right? Often in neutralization assays, we're seeing this. Yeah, I think usually they're working with virus that's passaged once, and the passage is from vero cells. I think that's what
Hmm.
we've seen most of the time. So, anyways, workhorse of cell type here.
Yeah, and from primary human cells. So these are ones that are taken from humans. Very so unlike viral cells, which have been in culture for maybe decades since they've actually
Yeah,
seen a monkey, but whereas this one is like directly from a live human. So
yeah. So
theoretically,
maybe in culture for just a few days.
yeah. So so it's supposed to be a
Um,
lot more representative of humans than other cell cultures.
Yes, yes. And they do them, and they do the human nasal epithelial cells at 33 degrees Celsius, right? Opposed to what we normally use in the incubators is 37.
Yep.
And that's just because it's on the outside of the nares. This is where they would probably grow. They're comfortable at this
Yeah.
temperature. And I mean, that would also be, it's also a type of a model, right? Like this is every cell culture is itself like some limited view of what happens in reality.
Mm.
And here they're trying to capture some temperature, right? Like maybe the virus behaves differently in this temperature.
Yeah. I mean, also like they use this thing where they have layers, so they say that they put like the culture so that it's um on a liquid layer so that it almost mirrors that
Yes. Yeah,
epithelial
the air interface
air
layer. Yeah,
interface
and
layer.
I
Yeah.
mean that is we've talked about this before, organoids, right? Or like things that recapitulate tissue structure. That's what they're doing here, right? So it's not just that they have cells that came from some part of the body and now they live in like this like nutrient bath, right? But they draw them up to the meniscus, and that's enough to start them differentiating in
Yeah.
some ways. Yeah, so that's actually what I did in my graduate school stuff, like was growing those types of cultures.
Oh wow.
And they don't show you any pictures here, but what's fun is that they have histology, right? Like you can slice those and you can see, right, that they look different, that they start differentiating a little bit. They don't even comment on that or show us what's going on. So like without knowing, right, like what that is, it's hard to say anything.
Yeah, a big sad face on that. They should have included some nice pictures.
Yeah,
But then we didn't get to
yeah,
see the. Oh yeah, quick disclaimer: we didn't get to see the extended data figures. So if they,
yes.
because we couldn't find them, but if
Yeah.
they did have these pictures, then they should share it with us so we can see them.
Yeah, I mean,
Yeah.
it's nice to know. Again, it's like just learning about how the model is because interestingly, in these two examples, they don't really see a difference, right, in the replication abilities.
Yeah.
But I guess actually maybe it's not that interesting. Is this, are they doing, they're not, yeah, they're not doing any sort of competition here, right? This is
No.
just the straight virus on these
Yeah.
cells. So it binds better to the receptor, we know from the biochemistry, right, in several different ways.
Mm.
But when you put them on the cells, they all grow up the same.
Yeah.
And so in some ways, this is more of a control, right? To just say that they're viruses because we know that this is a mutant that they've made in the lab,
Yeah.
right? They also kind of want to show us that that process didn't do anything for the ability of this virus to grow in these two
Mm.
circumstances. Mm-hmm.
And now they do different temperatures. So they go from 33, 37, and to 39 degrees. And
Mm-hmm.
they do start seeing differences as the temperature gets hotter. But
Yeah. I mean, it's interesting. These are also different cells, right? Now they've switched to bronchial
yeah,
epithelial cells. So it's another primary source. So they've switched the source, and that idea is that like we've tested like the nose, right? Like the open part of the nose, and then we've tested these monkey cells. But when we think about SARS-CoV-2, we're actually thinking about a lung
mm,
infection. And so maybe down there are the cells that we will see this difference more pronounced.
yeah.
Um,
So they go like 33 is to be like, oh yeah, it's kind of cool, colder. So and then they go into 37. That's
Higher up in the tract and lower into the tract.
and
Yeah.
then like 39 is if you've got a really really bad fever, then
Yeah, it's a pretty bad
like that's
fever.
very yeah, that's like you need to go to hospital. That kind of level of fever.
Yeah.
So
A little bit contrived, I would say, right?
yeah,
Because in my opinion, right, these experiments in cell culture, because um yeah, like the situation's kind of weird too, because it's cell culture. So like there's a lot of things that change in those temperatures and these locations. But nevertheless, they try to sort of make the point that perhaps, right, that it has some advantage in sort of the disease state that we see. Closer to the disease, there is a better replication change. Or yeah,
Yeah,
there is a replication advantage for the mutant.
yeah. So,
And that's odd, actually.
yeah,
I mean, to me, that's odd because replication is something that I would think is more dependent on like the polymerase, like
yeah.
stuff that goes on inside of the cell. But I guess also inside of a dish, it's a mini population.
Hmm.
So like if there is the ability to, right, infect more cells, transmit between the cells in a dish, you're also seeing that effect.
Yeah. I mean, it's weird
Yeah.
how like the actual like original form of SARS-CoV-2 seems to be doing much less well at higher temperatures. Uh,
Hmm.
so I mean, it could be so theoretically it could be it could be variants in the actual experiment itself. So we don't necessarily know that for sure, but it's an interesting.
Yeah, I mean, another thing that I could say about the temperature, maybe, and again, like all of this requires, these are all just like things we're learning from looking at a single graph. It would,
Yeah.
if they all could spawn their own spin-off paper with better experiments that confirm closer to the point, right?
Right.
But the reason I want to talk about it is because they talk about enhanced replication. And
Mm.
so, like, right, like if this is data that they're going to use to support replication, then we should dig in a little bit.
Yeah.
And what I'll say is that well, maybe the temperature lets it be more flex, like right. The temperature is keeping that open conformation, right? At higher temperatures, you have less like sticky binding between things,
Yeah.
and so with that mutation, like it's even more. Those RBD domains are just like flying all over the place, right? Like they're all questing, and yeah, with the regular one, like the that extra bulk, that charged amino acid, is able to hold the closed conformation better, and so it just behaves like normal.
Mm. So now,
Okay, but let's move on.
yeah, the competition assays. So
Yeah,
these,
because this, I like this because we're getting closer to the data that we saw back in May, right?
mm,
Like in May, the observation was, right, that like over over seemingly the course of this outbreak, we had more of this one virus come to prominence.
yeah,
And so I like this as a model for looking at that. They do a 50-50 inoculation.
yeah, a 50-50 inoculation that, according to the graph, is more like a 60-40 inoculation, but
Not perfect because
not
they got to
perfect.
measure out
They
these
never are.
small amounts of virus, right?
Yeah, they never are. It's almost impossible to get it exactly correct, especially with viruses where you only know the exact numbers like maybe a day after, and there's but
Yeah.
at that point it's too late to tell.
Right.
So
I guess yeah, it's important we talk about like we think about this is the great thing about going through figures, right? Is that you really think about that experimental design. They grow up the virus separately, right? They have cell cultures
yeah,
that are producing this virus and they purify off that liquid. They have to measure how much virus is in those things, right? Through some technique
yeah.
and then back calculate the concentration of the virus that they see and then drop those on, hopefully in 50-50 amounts.
Yeah, I mean, I had to do this with bacteria, and bacteria are a
Hmm.
lot easier because I had some reporters. Whereas viruses, you have to put that, you have to genetically sequence it. You have to analyze those
Yep.
sequences. So a lot of work. So it is nearly impossible to get everything absolutely correct.
Oh, no, I'll say that it's probably easier for viruses. Well, you had reporters where you used to figure out the concentration. Is that how you're saying you did bacteria?
Oh yeah, bacteria. I had like some uh, bioluminescent reporters, so I can tell like one of them glows, one
I
of them
see. So,
doesn't.
like, your bioluminescence was correlated to the amount of bacteria in a sample,
Yeah,
right? So, in this, they don't have any reporters in
yeah,
this, right? So, it's like they actually have to do plaque assays, I
yeah,
think. Or I guess they could do an infection and or they could sequence they could do the RT-PCR, right? Like genome numbers.
yeah.
But that wouldn't give them infective particles, right?
Mm,
Because there could be like right, I mean we've talked about this from the diagnostic side, right? It would give them like non-infectious particles. It's not good either.
yeah.
Uh, so anyways, basically it's not an easy thing to get 50-50. They tried their best. And they end up giving us more of the mutant in all these circumstances, right? As in terms of starting amount.
Yeah. So, I mean, they start off with a 50-50, then they do a 1 in 10 where they have just lots of mutant already, and then they have
Hmm.
like the opposite where they have very little mutant. And
Yeah, just to see if there's some effect, right, of the starting populations on this.
And the thing I want to highlight is also with competition assays, a really important thing is also the variance, because that variance is also an indicator of the level of founder effect. Um,
Mm. Hmm.
so, I mean, when I was doing this, the thing I'd like to do is have a kind of a third version of like a neutral, like founder effect control, where you'd have like something with an innocuous mutation that you could check as well.
Oh, yes, yes.
So you can actually quantify the amount of founder effect.
So a
Unfortunately,
missing
as
control here, perhaps.
yeah. Unfortunately, with my one, it went wrong because the thing that because I just knocked out a bit of an unknown part of the genome that actually made it look like it became more infectious. So, which just illustrates how difficult these sorts of things can get. So I can understand why they didn't do that because with viruses, it's very very hard to find any part of the genome that doesn't do anything.
Right, that they
So,
could do like a neutral mutation like that to do that control. Hmm.
but I mean, I think it's quite indicative that like when you see the kind of variance in the 110 versus 10 versus 1, that you can sort of see that there is a very big difference there. So you
Yeah, seeing that variance actually to me, like when they interpret this figure in the paper, they say like, oh, they see an effect, but to me, I don't see an effect when I look at this data, right? I'm like, I don't know how you can tell what's happening here, right? To me, it just looks like things happen.
well, I mean, I think the thing is like that with the 10 to 1, there is many more options for a virus that can outcompete its competitor. So therefore, you see
Yes.
that very much more variance in the number of the amount of it being present in the culture. Whereas if it can't
Yes.
compete, there are many fewer options. So there are many fewer universes where it's able to have increased in the amount of its own presence in the culture.
Agreed. Yeah. So it's kind of like when, yeah. So for me, it's like the 10 to the 1 in the direction of, yeah, the 1 to 10 in the direction of our mutant, right? Where there's like, or sorry, our, wild type. Oh no, our mutant, where there's tons more mutant, right?
Yeah,
And there's not a lot of that wild type. We rarely ever see, right? Like a data point that drifts down, right,
yeah,
where the mutant is one, right? So like that gives us a nice view. But then in the other two cases, we do see instances where the mutant takes prominence over the wild type.
yeah.
Yeah.
Even then, you can
Yeah,
sort of
so
see
I
an
agree. Yeah, in
imbalance.
that lens, it's much nicer to see. I think to me, I was just looking at the differences between the time points. I was like, I can't see a difference there.
Well, I
But
mean,
yeah, it's really important that they use these different starting inoculations
yeah.
to show us. Hmm.
And I think like even then, firstly, you can see an imbalance in the number of like data points above and below. And
Hmm.
also the thing that I have to like highlight is that even if you inoculate a culture 50-50, not all those viruses are going to cause an infection. A lot of them are going to ping back off, and you don't have, even if you try to do it perfectly, you don't have that control. Which is why checking these variances and the found effect can actually help you get an idea. I mean, if you're really, really clever, you can actually use this mix to calculate exactly how many viruses cause the initial infection in your inoculum. Like you can actually
Oh, sure.
calculate the size of that bottleneck.
Hmm.
They didn't do that here, and I can understand why because there's a lot of very different variations happening. But that's
Yeah.
that's why I was very interested in competitive indexes when I was doing them during my PhD. So this is
Hmm. Yeah, because basically, if you can collect enough data, right, and calibrate all these different ratios, then you could get a really good sense of like the frequency in which the event happens,
yeah
the
you
competition wins out. Hmm.
yeah. So if you're actually trying to put, so if you're doing like an infectious test and you're trying to cause an infection, it's really helpful to figure out exactly how much, what percentage of your inoculum is actually getting to cause an infection in cells. So
Anyways, let's move on and
yeah, let's
let's see. So they repeat these 50-50 competitions now with their bronchiolar um, epithelial cells,
yep.
and they see the same. They see effects.
Yeah, they see that again this competition. So the virus is literally fighting for ace 2 to try and see whether they can cause an infection.
Yes, yes. And then we check sort of a few days afterwards and see like which virus is prominent in the culture.
Yeah, and again that big for the first time this weekend, Orange is winning. So but
Yeah, I guess it's also nice to see here that it also basically there's a complex phenotype happening right in these cultures
yeah,
because replication presumably is also affected,
Mm.
right? As we saw in some of the growing in the growth curves. So,
Yeah.
and it does make an even more pronounced difference over the 5 days.
Yeah. I mean, it seems like they form. Looks like they're forming an equilibrium at some points, but that's again that.
Yeah, because like in days 3 to 5, basically, it's like stabilized a little bit.
Yeah. I mean, so it looks like it, but again, it depends on. We can always extend these graphs out further, and then you see a difference. So you never really know what's going to get fixed in the population unless you
Yeah.
run these
And
experiments.
another thing I'll say is that these are also these funny cell culture
Mm.
models, right? That may have been, I'm not sure if did they differentiate the bronchial epithelial cells?
I
I
think they
don't remember.
still use the air
Yeah, they're still using the air liquid
interface.
interface, right?
Yeah.
And so if they are, yeah, if they are using that interface, they say primary human nasal epithelial mucil air, uh, MBA. I can't see, but if they are using that cell culture, or I mean any
Mm.
cell culture, how many days, right, can you let that experiment go on for, right? There's also a limitation in the materials that you're using
True.
to sort of gather some of that data. Because I
Yeah.
know when I was working with them, like over time, like stuff would happen, and that may or may not be related to the infection that I gave them, right? Because they're just getting old.
Yeah.
That's like a big part of it. Yeah, because like in these cell things, like there's no circulatory system, like there's no upkeep on those cells.
Yeah.
And because they're primary, there may not be that stem-like, right? They don't have that like infinite well of replication that cell culture cells do,
Yeah.
right? Because they're crossed with some weird ass cancer. Yeah,
So yeah, that's a very very good point.
limitations on the model,
Yep.
always.
Every model has limitations.
Okay, but from those competitions, like I think we're getting, I do think that those data are really helpful in informing, right? Like a nice biological reason,
Yeah.
right? Uh, why we might see the increase in this virus in the population is because, in the small scale and cell culture, it outcompetes. It outcompetes.
Yeah. So we've been talking about cell
So,
cultures and that kind of model. So let's move on to a mammalian model with
mhm.
mice.
Yep. Yeah, let's increase that complexity.
Yeah.
So we have our mouse model. This is the diagram in which they show us the horrible things they're going to do to this mouse. Where they infect them at the beginning intranasally.
Hmm.
They take swabs across this 4-day window.
So it's really
Uh,
tough to swab a mouse because the nostrils are. I mean, when during my PhD we tried swabbing mice, and the only thing we could get is like suture material, which is so fine.
yeah.
But usually with throat swabs, they
Oh, tiny sutures that you like put in the
yeah.
nares. Yeah.
In the end, like we just literally dunk the mice's noses on the agar itself and just swab the mice across it. Um,
Sure.
but yeah, the throat
Little
swabbing,
streak plate with mouse nose.
yeah, it was really sensitive, but it was just yeah, it was actually a really sensitive for the bacteria that I was working on. But I wouldn't use it on viruses. I don't think that'd be.
Sure,
But
sure.
But you can do like throat swabs where you've got like this cotton and you just have to swab the back of the throat on the you, I can't I used to remember the name of it, but like where the nasal cavity opens up into the back of the throat and
Hmm.
swab the secretions from there, or even
Yep.
like salivary secretions, and yeah
I mean, I think isn't that what oral that is oropharyngeal
yeah
is where the yeah is where the mouth hits the pharynx,
yeah
right? So where your nose and your mouth connect
yeah.
to go down the pipe. Yeah.
So yeah, I mean the thing is like if for humans it's you get much more because we have our tonsils back there because we're not
Hmm.
obligate like nose breathers. Mice only breathe through their nose.
Right. Right.
So there's so it's a slightly different level of sensitivity you're gonna get from that, but
Absolutely. And
it's
I mean, I think they know that because they're also planning in this plan to sacrifice the mice
yeah
on certain days and harvest their organs. You get much more reliable results from just the organ
yeah
harvests. Hmm. And so they use a human knock-in mouse, right?
Yeah,
We've talked about this before. I think in the previous time we talked about this, we were talking about infecting mice with adenovirus that contained human
yeah,
ACE2, which is nice because it's like quick, right? You just like put in this ACE2 beforehand. But here they actually go through the effort of making a stable knock-in.
yeah. They because this is something that would take a very long time too, because they bred several generations of mice. So they
Yes,
started out
yes.
with like homologous recombination. So this is where they have a short section of the gene they want to change on their like kind of plasmid that has all the genes they want to put in, and then
Hmm.
like they give it to cells, and every now and again cells when they're replicating can get confused and start replicating the plasmid stuff into there.
Yep,
So incorporating the cell, and they put that cell, the stem cell, into a blastocyst. So that's like earliest stage of replication,
yep.
and you put it in, it
I mean,
sneaks
this
in.
is IVF, right? This is
Yeah,
IVF. This is like the the sort of stuff that people always talk about in science fiction, right? Where it's going to lead us to some crazy dystopia of editing embryos, right? And we do it to mice all the time for research.
yeah. I feel that doesn't show us in a great light, unfortunately, but yeah, no, they do it in a long way with a lot of breeding and a lot of like and so they have this really stable knock-in, which
Yeah, and I guess like what was nice about this model is that it's not going to make any ace. It's not going to make any mouse ace two,
yes,
right? Like in this version. So like it's very clean experimental model to work with.
mm,
I think I even read that they use the promoter sequences and the regulatory sequences of the mouse
yeah,
in this. So right, it's not there's no human regulatory. The only thing human in this mouse is the ACE2 sequence itself.
yeah,
And that's great because that is the sequence that the spike protein likes to bind to.
yeah, and they do test their model out to see whether like
Hmm.
SARS-CoV-2 infects it, and they find like a log for a, I think it looks like 100-fold difference between
Yeah.
the ACE2 knock-in and the wild type, which is
Yes.
really big.
Yes. So yeah, a nice figure to show us that it works. And they are going to use this mouse model to do their challenge experiments again. So instead of on cell culture, we're going to do it right in the mouse nose, nares.
Yep.
We're going to try to give them the best 50-50, and then we're going to watch them and also harvest organs.
Yeah. And so that is what we see.
Hmm.
So
A huge, so much more variance here. These are swabs too, right?
yeah,
And so, like as you said, like some of these swabs, they just might not be good, right? Like
yeah.
that one, K8, day 3, like that's probably just a bad swab,
I mean,
is my thought.
it could be, or it could be that different areas of the mouse nose have different levels of the virus, and because there
Right.
is a lot of like, because
Well, that's what I'm saying. Like that would be considered bad swab technique, right? Because like you assume every swab is supposed to give you a good sample of everything.
yeah, and
Yeah.
you don't. So I mean, thing that you used to do is like not just the one swab, but several swabs, and then
Gotcha.
compare them.
Hmm.
So
Hmm.
I know I tried doing that and try and then take an average to get an idea of the variance of that you'd get in your
Hmm.
sample. Uh,
Hmm.
but yeah, so I mean, I don't want to go into the so because again, like with this, like you're not just looking at like one uniform infection with the mouse. Like, do these different like kind of viruses will be at different parts of the nose potentially?
Hmm.
Because in fact,
Yep.
like if you giving them, they gave them 20 microliters, so some of that would literally go down into the lung as well. So
Yes.
and there'll be different
Yeah.
patches of these viruses. They wouldn't necessarily be directly competing for a while, or
Hmm.
so a lot of it does depend on those founder effects. Um,
Yeah. Yeah. But that's something too that that's interesting because like that's going to come out in I guess maybe it'll come out later because when we get infected, right? How much virus really gets in us? Like we don't get we don't
yeah,
get um, liquid poured into our cavities, right? I mean, hopefully we're not getting that as our infection route. Um, yeah. So again,
yeah.
limitation of a model.
Yeah, I mean it's an interesting. I mean, because I know there's this idea that face masks reduce the dosage of virus, so it makes those sorts of. So while it doesn't stop those contacts from happening, it means that there are going to be less successful versions of it.
Yes.
Um,
Yes.
so
But I guess the takeaway from this is that it does recapitulate what we've seen in cell culture,
yeah,
more or less. Yeah, the
I'm
patterns aren't as clear to see, but Yeah.
yeah, it is a lot messier because again, also got the immune system to compete with as well, and
Yeah.
that can have a strong can increase the variation of what you
Yes.
see because
Yes.
again, you
But very promising, right?
yeah
Promising to see that we're seeing some recapitulation of this more complicated model.
yeah
And maybe if we want to get better resolution on that, we can go and chop up right? We can homogenize some organs and get counts from that. And
which
that's what we're
is
seeing in 2D.
yep
And again, we're still seeing that competitive advantage pulled through here.
yeah and you can see it in the majority of mice, but obviously there is still variation. There is that founder effect, that sampling effect that is affecting what we see here.
Yes, that could be influencing this. Yeah.
Yeah, but
But I mean, what's interesting for the different organ systems would mean that like that effect would also be like it has to hold true even against another organ, right?
yeah,
Like, yeah. So it's like we're even seeing more data points in that way, right? Like covering more of the body, and seeing like what got there.
but
So
yeah,
is it possible that in every circumstance it was just chance that
yeah,
this virus got there first? Or, right, is it, as they're hypothesizing in this paper, that there's something about that virus, its increased binding affinity towards ACE2, that's driving the differences?
yeah. And here it seems like the trend clearly shows like orange is going forth. Like
Mhm.
the new version of the virus seems to be in most of the cases outcompeting the original form. And it
Right.
does, and it's seeing like kind of stuff like where these random things, that's not something that's expected, and it'd be weird if we didn't see outliers in data. So
hmm hmm Yeah, I mean, again, I think that this is where they're showing us the experiment they did, right? They're not
right.
hiding those things. And it's not about finding that flaw and saying that if you can't explain the flaw, right?
Mm,
You can't explain the outlier, then that's where it becomes, well, is this experiment good or not to
mm,
tell us what's going on? But we can see it, and like they're showing us multiple lines as well,
mm.
right? To try to support this hypothesis.
Yeah, and is the next one is let's
Yeah,
see,
so
yeah,
they're gonna give us the whole like circus of animals here,
yeah,
animal models. Um, they want to relate this to transmissibility,
mm,
right? So far everything we've seen is about competition. Competition that they tried to link to replication.
mm,
I think a bit shakily, but like, okay, whatever.
yeah,
They're linking to replication. But they also have this idea that it could also just transmit better. But
yeah,
this model that they're doing isn't about transmissibility. So they switch models, and we're going to Syrian hamsters,
yeah.
which we sort of thought we talked about the hamster transmission model.
Yeah, we've talked. I think
Hmm.
like we and last week we talked about the evolution of like ACE2 and how
Yes.
it's very similar in hamsters and humans,
Hmm.
therefore making
Hmm.
hamsters a much more uh, human model compared to mice.
Yeah, they don't have to do any genetic engineering to these hamsters to have them be infected.
Yeah,
And so in this type of experiment, they can inoculate one animal and then with brazen disregard for social distancing rules, bring them into close proximity and see what that contact, how quickly or and what strains that contact ends up acquiring.
yeah, and again, this is. I mean, for for me, like in terms of like looking at competitive assays, this is where it gets very. Where I would love to have seen graphs showing like the competitive ratios because then you could sort of see like exactly like because you'd hope to see like massive variation, but if there's a very small like infectious dose, but these hamsters are in each other's like business all the time, so
Hmm.
and it is
Hmm.
interesting how you can. And again, this one is like a clear difference in D614G. I mean, one of the things
Yep.
that would be very interesting for me is to see what that founder population size was and see how much difference that would make. But um,
So that means they would have to, what you're asking for is like seeing them try different inoculums.
uh, yeah, that's well, it's inoculum
Like, how would you get
mixes.
at the data that you're speaking about?
So yeah, I'd say the inoculum mixes to like see
Hmm.
like how the shift between them.
Yep.
So if you have like a 10-1, what kind of shift would that cause in the ratios? And then
Yep.
use and like also to see like the degree of variation. But from
Yeah. absolutely.
then again, from what I'm seeing here, it seems like that there isn't much variation. That the competitive advantage D614G gives is so big that you probably wouldn't see that effect if you did those kinds of experiments. Because
Yeah, yeah. I mean, yeah, in some because they also it's like it's the second day, right, in
mm,
which that they bring the hamsters together.
yeah,
And so in that day, right, from the swab that they get, already the D614G right mutant has taken over.
yeah,
Mhm.
yeah. I mean,
At least in the first three pairs. I guess the next, oh yeah, in the next three as well, right? In that within that first day, it's already taken over. And actually, then all these circumstances, the the contact gets it immediately after meeting their infected buddy.
yeah. I mean, this is something that I mean, I always have to be concerned on when because I had to do a similar thing when I was infecting mice for transmission studies, where if you have to keep the
Mhm.
mice safe for a day because you're worried that the inoculum is still on their faces. You need to make sure
Right.
that it's actually causing it's actually proper transmission and not this the fact
Yeah.
that the
It's
mice.
not just like they were able to aerosolize some of the liquid that was already there. This is like virus that is being made inside of these animals. Yeah, being
Yeah.
transmitted through the
And
air.
if I recall correctly, Syrian hamsters aren't like as social as mice are. Mice, when they are in a cage, they bunch up together. Syrian hamsters, they tend to like their own space a lot more.
Right.
So, um, that,
Well, I mean, I mean, if the previous paper where we talked about this model was any evidence, any indication of how they did this study, is that they just end up like they put two smaller cages right in the main
yeah,
cage and force them to be co-housed in a close proximity.
yeah. So, this one it seems like they just put them in the same cage, and so it wasn't
Oh,
just
they put them in the
airborne.
same
They just
picture.
so they could it could have been I mean, they weren't looking at airborne transmission, so that's not really too much of a problem. This could have been transmitted by fomites or even through I don't know, maybe grooming potentially. I'm not sure.
Yeah, do they like touch each other's noses together maybe? Yeah. But this is what
Maybe.
you're saying, like you're not sure how this translates into hamster behavior, right? Like they may
Yeah.
not be doing some of those behaviors.
Yeah.
It's all in the privacy of the cages here.
Hmm.
We don't have the videos.
Yeah.
We don't have the video evidence to tell our viewers.
Oh, but
Okay, so that's one transmission model,
yeah,
and they try again with ferrets.
ferrets. Yeah. So because ferrets are a much recognized transmission model for food and for and I think they also 'cause they're not quite as closely related to mix because apparently get SARS-CoV really badly, but um, like for instance, again they use an influenza model because they got a similar system to the humans. and and this is actually really interesting. I mean, the first ferret that they experiment on has very interesting results because in
Yeah,
that way,
the other virus comes to prominence in that one. Uh,
and the other ferret doesn't get the virus,
mm-hmm, mm-hmm, mm-hmm.
which, which is yeah, that's
Which
just
is really
almost
great
like
confirmation, right? Like it's a
yeah,
nice
yeah,
confirmatory, yeah, thing to happen.
yeah,
I think there's another instance in pair four where the other ferret doesn't get the virus either.
yeah, that's
Um,
true.
and the way that they explain that one is they think that it never gets up to a high enough titer, right? Because that
Mm.
ferret actually ends up controlling. Actually, all these ferrets, oh, seem like no, not all of them, but that ferret is definitely on the trajectory to control the disease because they have the total copies per mil as the numbers underneath, and in pair
Mm.
four, that virus is decreasing rapidly.
Yeah,
Mhm.
so that's actually an important thing because the amount of virus in the air affects how much it transmits.
Yeah,
So that in itself so the ferret data again adds a lot more complexity and doesn't necessarily isn't necessarily while it looks like there's some great stuff here, it doesn't compared to the hamster data. I feel like the hamster data really sold their story a lot more as ferrets. It seems like that SARS isn't as great at infecting ferrets as it is at hamsters.
yes, yes. I think that I mean, and we didn't cover any ferret transmission model papers, right? But like
Yeah.
when we covered the hamster one, they were I thought they made a strong case to say that this hamster model works, right, as a
Yeah.
transmission study. So like, yeah, I don't know where this ferret one comes from. We just haven't read that paper yet, but maybe it is a weaker model, and maybe they use it. Maybe it's showing us something else. But I don't know what it's showing us actually. Just like if the virus is not as good as infecting, then it doesn't transmit as well. Okay, well, I think we
Yeah.
probably would have known that before we did this experiment. I think more the times that they do see the transmission is a nice confirmation on the hamster data, right? To say, okay, even when we swap out the ACE2, right, for another one, a different immune system and a different receptor, we're still sort of seeing we still see the some of the evidence, right, that this is the
Yeah.
case. Yeah.
I mean, in the ferret studies, the original version just never transmits, as far as I can tell.
Yes.
They, so I think that
But
it
also, like,
shows.
I would say that in the ferrets, the mutant version does a lot worse. Like in the pie graphs, there's a lot
Mm,
less blue, except for the one case where it does.
yeah, yeah.
um,
The
which is interesting. I mean, that may be like a found, right? Like, is that like a founder sort of thing, right? Like, uh, yeah, just
it,
seeing like that sweep of the virus population. Yeah, I'm not
I
sure.
mean, it very well could be, and because again, there is if there's a strong immune response and it doesn't get infected, then you're going to see a strong selection effect that's going to
Yeah.
really randomize things. So if the ferret, if like say on day like 4, there's a massive immune response from the ferret that
Hmm.
clears out a bunch of viruses but only leaves a few, that's going to induce a second bottleneck that
Hmm.
will again induce more variation. So
Hmm.
that could be one of those reasons why you will see a lot. Uh, Again, why like kind of doing these kind of mixed assays can lead to some really interesting outcomes.
Yeah,
Um,
absolutely. Absolutely. Um, I don't, they never say whether or not these ferrets will be sacrificed in the end. Uh,
no, I don't think they do.
yeah.
I mean, I'd like to think they didn't sacrifice them in the end
Yeah,
because it looks like they all survived.
yeah, and they clear, right? It seems like they're
You,
not doing so poorly. Like, right, this is not
yeah,
a, yeah, this is not a, even in the hamster model that we talked about back then, those hamsters didn't have to be sacrificed at the end because they mostly clear that infection.
yeah,
Um,
you wouldn't get any data. The only
yeah.
issue would be that the safety concern that your kind of managers will be telling you about, where just because it's got infected with SARS-CoV-2, we can't let them out.
Yes,
So
yes, yeah. What to do with them now? Yeah, exactly.
yeah, so that's the main concern, and seeing like how biolabs deal with that.
Hmm.
Um,
Hmm. Yeah,
so
but
because I
I guess more I was speaking about they don't show us any of that could be something that they might be able to show us maybe some of the stuff about the disease model itself, right? Like what did the
yeah
disease look like on the day that right the transmission occurred or like at the end of the course. I guess that's tough because like you can't you can't do that without stopping this timeline, right? Like you'd have to take one of
mm,
those pairs and on a day sacrifice and observe the tissues. And so like yeah, that doesn't fit
yeah
in this in the
and
amount of animals that they used. Hmm.
yeah I mean you lose power every time you do that. I mean I guess one thing that would be interesting to see is like the antibody responses or the immune responses to
Yeah,
the
take some
virus.
bleeds maybe, take some bleeds and try to see
Yeah,
what's going on. Hmm. Oh, I mean bleeds and some flow cytometry, but again, like we're looking at totally different immune systems here. So
oh yeah,
you'd have to have some good, I think the data would be interesting, but also difficult to interpret. Like you'd need some really good sort of
yeah,
comparative immunology sort of stuff going on to make those, yeah,
yeah.
of that.
And like just from a practical perspective, you're dealing with much less blood because
Hmm.
like,
Hmm.
and that way you get a lot more variation and a lot. It's a lot. You have to be much more careful with how you handle your samples
Totally.
because, yeah. But I think that brings us basically to the end of this paper.
Yeah.
Um,
Yeah. I think that the best way that I categorize this paper in my head after reading it is sort of the last sets of data, right, from a long story, from the identification of this mutation down to some pretty good hypotheses with support, right, about the function of that mutation. And maybe an important thing to remember is that they never look at this mutation. They're not looking at it in models of increased disease. That's not what they saw, right? Like increased in serious disease, right, or like adverse events. That's not what they saw when they initially found this mutation in May, and that's not what they're following up on here. It's really that makes a lot of sense in the evolutionary scale of things, right? Like this wants to transmit. I mean, it's been very good at doing that. Um, and so yeah, that's how I'm categorizing this. A nice end to the saga of this particular mutation.
Yeah, I mean, I'm based. So, I mean, I think this is quite interesting because of the transmission aspects of it. Uh,
Mhm.
the Yeah, and also yeah, we have been mapping the entire saga of how this mutation came to be, and honestly, there's not really. The fascinating thing is like how it can change the way it transmits without changing. So the immune profile, as far as we can tell from all the other studies, is exactly the same because this
Hmm.
actual mutation is really right up inside the. So it's not exposed to antibodies. It doesn't change that. So it doesn't change the immune system. It just changes how well this functions as a molecular machine. Um,
Yes, yes, yes. And I think importantly, people are concerned about whether or not this will impact vaccines. And we've seen some papers coming out saying that they don't think there's a big effect on this.
hmm,
Because a big thing to remember is that when the vaccines were being made, phase one, right, phase two, right at the beginning, they were using early isolates from the Wuhan region that may
yeah,
not have had this mutation.
yeah.
So it is important that we know that our vaccines work, but as you said, the location of it sort of means that it probably won't be something. And we have seen some data saying that the same antibodies still bind to this variant.
Yeah. And so, from the perspective of like seeing how this works, though, I think this has revealed a bit about because I think that open combination will lead to some interesting ideas of like how proteins kind of change and how they bind. Because I think that's
Sure. I mean,
yeah,
from yeah, you're saying on the basic research side, we're kind
yeah,
of like learning about, yeah, like it's nice to have that story as illustration, right, of like what you can predict from
mm,
seeing a change, right? Because that's something that comes out from a prediction, right? You see that it removes this group that normally links these two subunits, right? And then it does, you can see that effect. This is the story of it. So the next time you see something like that in nature, you can feel more confident that your hypothesis is a good one to go down, for sure.
yeah, yeah. I think we've basically covered everything we can on this paper, and
Yeah.
it's nice, and it was a nice, short, compact paper that compared to like some of the other papers we've had. So it's been quite chilled out to talk about.
For sure, for sure. Yeah, so if anyone still listening along is interested in seeing us dive into other papers in this fashion, then please leave us your comments using the hashtag or adding our account, um, or I guess in the comments below as well. And we'll see you next week.
Yeah. See you next week. That's bye bye from now.
See ya.
See ya.